So, today we are going to speak on one of the important topic under the SWAYAM course of genetic engineering and recent advancement, and the topic of the today's lecture is restriction modification system. So, first of all, it is about the restriction modification system, and before understanding in detail about the restriction modification system, let us have a brief introduction that what type of the systems are restriction modifications. Basically, we can think this kind of system for a kind of defense mechanism which is ubiquitously present or occur in bacteria, not only in the eubacteria, but it has also been reported in the archaebacterial group of system. The restriction modification system is kind of extremely diverse group of enzyme which is having two major component. Of this, one important enzyme is the restriction enzyme and another important is called as modification enzyme, that is basically the methylase enzyme. So, in this particular RM system we are going to talk about two important enzyme. The first one will be the restriction enzyme and second important enzyme will be the modification enzyme, which is also called as the methylase enzyme. And we think these two important enzyme as a kind of a modification or a restriction modification system, and it is also an important defense mechanism which has been reported and has been observed in many of the bacterial species. And why the system is present in such bacteria? So, that is very important to understand, because it is naturally present as a restriction modification system which is a diverse group of enzyme, and they are ubiquitously present in the bacteria or in eubacteria or in archaebacteria. So, why the systems are present, why the bacteria use this system, we have to understand. So, the function of this system is to prevent the infection. So, it will prevent any infection of bacteria of invading genome or invading foreign DNA material. As we know in the nature, if bacteria are there, more than the bacteria, bacteriophages are there, and these bacteriophages are called to be the eaters of bacteria, they can kill the bacteria. So, mostly in these lambdaphages, what is actually the lambda bacteriophages, they infect variety of the bacterial species, they consist of the DNA material as their genome. So, the function of this system on infection, it will restrict or it will prevent or inhibit the foreign or the invading genome to hijack the host cell of bacteria. And how this system works we will see in the upcoming slides. But before this, we should understand this is the one of the important system. We can think it as a kind of defense mechanism which is present in the bacteria, and on infection with such type of foreign DNA, it will inhibit or it will restrict the, or we can say it will kill the foreign DNA. Any time the bacteria is exposed or any foreign DNA is introduced to the bacterial cell, the bacteria can restrict or it can damage or it can cleave the foreign DNA. That is the reason how it will restrict the foreign DNA to grow inside the bacterial cell, and at the same time it will also modify its own DNA. It means it is protecting the DNA. Basically, the restriction enzymes are kind of endonucleases enzyme. So, there are two enzymes here we are going to talk. One is the restriction enzyme which is called to be the endonuclease enzyme, which will cleave the DNA material, that is any foreign DNA material, and it will here it is restricting the foreign DNA, but at the same time it is protecting its own DNA. So, there is a very good discrimination between the self DNA and non-self DNA, which is kind of foreign DNA. So, it is protecting its own DNA from the endonuclease enzyme through the modification, and that modification is called to be the DNA where the DNA is used as substrate and methylase enzyme will act upon it and it will modify the DNA exactly on those sites where the foreign DNA is restricted or foreign DNA is cleaved. So, on infection the host is protected, whereas the foreign DNA is restricted by the endonuclease enzyme. Two important enzyme activity which is going on together. So, restriction enzyme is accompanied by the modification enzyme. Two types of enzyme, one is targeted towards the foreign DNA and another enzyme of this complex is going towards the protection of self DNA, so that the endonuclease will not attack, it will not fragment the DNA of the bacterial cell. So, bacterial cell have evolved this mechanism to protect themselves from any invading foreign DNA, and particularly here we are talking about what foreign DNA is basically the phages or the bacteriophages or the lambda-phages, which carries this DNA, and once they infect, you know that the phages can infect the bacteria and they are able to lyse the bacteria through this DNA. Once the DNA is introduced, the host cell is completely hijacked and the host cell is finally killed because of the lambda-phage infection. So, we know that this is a kind of primitive immune mechanism, like it can be observed like a property of a immune system what we see or observe in the higher organism. Similar to that, the bacteria also have this immune system, which is kind of the primitive immune system we can say, and because it can better differentiate or discriminate between the foreign DNA and the self DNA, and it will kill or restrict only the foreign DNA. So, the primary function of this restriction enzyme is to identify and target the DNA sequences. So, you know that enzymes are highly specific for their reaction, and being the endonuclease enzyme, they will cut the DNA through endonuclease activity. It is a fragmentation of DNA, and as it is an enzyme, it will identify the specific DNA sequences. So, they will target the some specific DNA sequences which are known to be the recognition site or restriction site. So, here what is very important to understand how this restriction enzyme act, what is its specificity, how the methylase enzyme act, and why the cognate enzyme activities are required together, and together we read or we understand the whole system as a restriction modification system. So, the primary function of the restriction enzyme, if it is a endonuclease enzyme, it will identify and target some specific DNA sequences, and these sequences which are targeted, which are actually cleaved at that particular site, the DNA is used as substrate for this enzyme and they act on some specific sequences, and those sequences where the enzyme binds and it cuts the DNA, it is called as recognition sites or the restriction sites. Now, we will see here the two important, as I told you, the restriction modification system is having the endonuclease activity. So, we will see the first enzyme is the restriction enzyme. Restriction enzymes are endonuclease enzyme, and it will cut the foreign DNA, but it has to also look after its own DNA. So, endonuclease activity is towards the foreign DNA. Being the enzyme which can cut the DNA, it is called to be the nuclease enzyme or endonuclease, because it is going to cut the internal bonds of the DNA material or the DNA substrate. The other enzyme is the methyl transferase enzyme. So, here it is very important to understand the next enzyme, right. This is called as methyl transferase enzyme. So, what is the role of methyl transferase? As the name itself is suggesting, it will transfer the methyl group to the DNA sequences, and it will protect, means it will modify the DNA bases or the DNA nucleotides of the host DNA, so that the endonuclease is no more able to recognize the same DNA sequences in the host DNA. Because when the endonuclease activity is going on, it is like it is a very well high certainty that the same kind of sequences, many of such sequences are also present in the host DNA or in the bacterial DNA. So, bacterial DNA has to protect its DNA from the endonuclease activity when it is on action. So, to do that, the methyl transferase activities accompanying the endonucleases, before the endonuclease starts its action, the methyl transferases activity, it will do the protection of host DNA from, so that it cannot be cleaved by the restriction enzyme. So, this is methyl transferase is basically, it is the transfer of, it is a methylation process. It will transfer the methyl group from S-adenosyl methionine to one of the important bases, like you can see that methyl transferases will modify the bases, it will basically modify the base. So, the base will not be recognized, the same sequence if it is present what the sequence is identified by the endonuclease. Suppose the same sequences are present in the host DNA also, but what will happen because of this methyl transferase activity, the bases are modified, they are modified bases. So, as we know the enzymes are highly specific, once the modification is carried out, then the endonuclease is no more able to identify the same sequences for its activity in the host DNA, whereas the foreign DNA remain unmethylated. That is the reason why only the it will restrict the foreign DNA, and whereas the host DNA is protected. So, restriction enzyme and its cognate or the counterpart is the modification system. So, they work together, and that is why they constitute together the restriction modification system. So, they work together in bacterial system. So, here we will see that this is like activity, it has been shown here in the restriction enzyme, that how it will cut. So, you can see I will tell you about like what will be the normal the sequence length, right, how many base pair normally it is recognized by the restriction enzyme. So, it will recognize the short palindromic sequences, right, very short palindromic sequences will be there. You can see the first one is like a stretch of double stranded DNA or foreign DNA, and restriction enzyme is cutting that particular sequence. So, you can see here it can be a sticky end or it may be a blunt end, and it will cleave the phosphodiester bond between the bases, between the nucleotide bases. The phosphodiester bonds are cleaved, and it will leave the 5S end and the 3S end open. So, basically it is going to fragment the DNA. Otherwise, you know, this is the foreign DNA of the phage. If it is intact, if it is present, it will start the synthesis of viral specific protein, and that will start the replication of the virus, and if virus replicates its DNA, then there is a like it is a irreversible process. It will ultimately it will start the virion synthesis. The virion synthesis means it will start the viral protein synthesis, and totally it will start the lysis. We can say that there will be a complete lysis of the bacterial cell. So, before the virus DNA is able to start its own activity, restriction enzyme will sense, it will recognize that specific sequences, and it will start its endonuclease activity, its endonucleolytic activity. It will identify them some specific base sequences, which is a short sequences, right, and it is a palindromic sequences. If you read it from the opposite side, the sequence of the nucleotides are same, and that is why they are called to be the palindromic sequences. So, oftenly they identify the palindromic sequences, they recognize the short sequences, and they will cut exactly, that is the target sequences for the restriction endonuclease enzyme, and you can see it is like a scissor. It will cleave that particular sequence and it will fragment the DNA, and the other after the once the restriction enzyme act, there is a fragmentation of the foreign DNA start, and further the other endonuclease or nuclease enzyme will completely break down this DNA strand or the foreign DNA strand. So, here you can see that this is the two important thing. This is the first part we have seen just now, it is about the restriction system, and second part is the modification system. So, what we see in the modification? So, first part is toward, it is targeted towards the virus, towards the phage DNA, towards any foreign DNA, and we are talking mostly what will be the foreign DNA. This is the what the mostly the bacteriophages. So, this has been observed mostly in case of bacteriophages. So, it is the bacteria has to protect itself from the phage attack, and phage releases the DNA material to the host cell, and then it starts the overtaking of the host cell machinery, and it will kill the cell. So, host cell is here is the bacteria, it is protecting itself. So, first enzyme is the restriction enzyme which has done its work, and what the other cognate enzyme that is called to be the methyl transferases, what it is doing. So, there is a unmethylated DNA of the host cell, right, and the same sequences like the lambda phage DNA or the foreign DNA, there is a probability that the same type of many such sequences certainly will be present in the bacterial genome or in the host genome also. So, it has to protect its own DNA from attack by the endonucleases. It will be like a self destruction. So, to protect the self destruction, what the bacteria is doing? It is synthesizing one more component of this modification, that is called to be the means you can say it is a methyl transferases. So, before this you can see there is a unmethylated DNA, and now you will see that methylase or methyl transferase enzyme is coming and it will modify the bases. So, methylation or the methyl transferase is a very important activity of the restriction modification system. This is called modification. Why? Because the DNA sequences are modified. Here what it will be done, it will take the methyl group from S adenosyl methionine to the C5 fifth carbon of the sugar of the DNA, or it can be the N4 amino acid of the cytosine or N6 amino acid of the adenine. So, this is some sequences where it will add the methyl group, and once the methylation is done, it is called to be the modification. And with this modification, the restriction enzyme will not cleave this methylated DNA. So, it can cleave only, it will restrict or inhibit only the unmethylated foreign DNA, whereas the host DNA is modified through this modification and it will not cleave the host genome. So, this is how the two system works together. Now, these are the important things, like we have to understand what is the recognition sites for, you know that the DNA, your DNase enzyme which is called to be the restriction enzyme or endonuclease enzyme, they recognize some certain important sequences. They have their recognition sites in the DNA and they are highly specific towards their recognition sequences. Once these sequences will be present, this enzyme will recognize and then it will start its action. So, these sites are called to be the recognition sites, and we have to understand, as I told you, they are very short sequences. It is around 4 to 6 base pairs, and these sequences are very often you can see they are the palindromic sequences, means that they can, once you read the same sequence on both the DNA in opposite direction, the sequence is same, and that is the, you can see that is the particular sequences which act as the recognition sites, these palindromic sequences which are just 4 to 6 base pair in the length, and the restriction endonuclease will do the double strand breaks and will start the fragmentation of the foreign DNA. So, this palindromic nature of the recognition sites allows, this is the reason how the restriction enzyme can bind to the DNA in highly specific manner. So, you remember, being an enzyme, being the DNA as a substrate, the restriction enzymes, how they are specific, because they recognize their recognition sequences and they bind to these sequences only for the cutting or the cleavage of the DNA, right. So, it is a highly specific cleavage and it will ensure that it will precisely and targeted cleavage of the foreign DNA is only observed in case of restriction enzyme. So, here you can see the very good diagrammatic illustration that how this defense mechanism is found in the bacterial system. It is just like a property of the immune system present in the higher organism like in us. They can be observed as a primitive immune defense mechanism. So, you can see the phage is binding on the bacterial host cell, and then as soon it releases the DNA, recognition sites are identified, right. So, here you can say that recognition sites are identified by the REase enzyme, or we can say the restriction endonuclease enzyme, and it will start destructing the foreign DNA which is unmethylated. On the same side, the methyl transferase component of the modification system, at the same time it start modification of the host genome, so that it will protect the host genome from the REase. So, here it is a very good example of the defense. It is a very good discrimination between the self and non-self DNA, and methylation is only in the self DNA, whereas the unmethylation is there in the foreign DNA, and the methyl transferases will do this discrimination. And you can see here the bacteriophage's DNA are in the they are now targeted by the REase enzyme or from the restriction endonuclease enzyme, whereas the methyl transferase enzyme will protect it. So, it will ensure that this enzyme will have the good ability to discriminate between the self and non-self DNA. So, it is just like how it is ensuring the protection of the host genome from the endonuclease activity, and the REase is only directed towards the phage DNA, which can be said as a invading genome, which can be a foreign DNA, which is a phage DNA. So, they are like defending their own self from the phage attack, the DNA has been introduced from the phage, and we can see that it has been firstly observed. This particular phenomena has been firstly observed in the year 1950s. They have found that some of the bacteriophages are not able, or they are poorly growing in the bacterial cell. So, they were thought, what is the reason why the some of the phages are not able to grow in the bacterial cell, and the reason was the presence of the several type of restriction enzymes. So, they found that this kind of restriction enzyme are restricting the phage growth. So, this is how the discovery has been started in the year 1950s, that they found that some of the lambda phage DNA are poorly growing in the bacterial cultures, and they means these systems are restricting the growth of the phage DNA, and that is why they are not able to grow in the bacterial cells. And slowly we see that yes, there are various such restriction enzymes are present, which are like a bacterial defense system, and they are protecting the bacteria from invading genome, or we can say any foreign DNA. So, this is how we can say that when the bacteriophage or any other foreign DNA carrying the harmful genetic material invades the bacteria, the restrictions enzymes are activated. So, it is like a summary which I just now speak about, that is like how the restriction enzymes are activated once they find any foreign DNA is coming, and they identified their specific recognition sites within the foreign DNA, and they bind to them, then they start cleaving the DNA at specific phosphodiester bonds, and they will start the fragmentation of the foreign DNA. This enzymatic cleavage leads to the complete fragmentation of foreign DNA into smaller non-functional pieces, rendering it harmless, right. And they at the same time, however, they also prevent the self destruction, because bacteria also must ensure that its own DNA remains unaffected by the restriction enzymes. So, they are also seeing that the restriction enzyme should not start the self destruction, and that is how the modification are ensuring to prevent the self destruction by the restriction enzyme. So, to achieve this cell protection, the bacteria employs a protective mechanism, as I said just now, that it will involve the addition of methyl groups to the specific bases in its own DNA, which is called as DNA methylation. And we should, the bacteria is also so much clever that this methylation modification is not going to interfere with the DNA base pairing or any other function of the DNA. So, this is that few specific bases are only modified or methylated, so that the restriction enzymes are blocked here and they are not able to attack on the self DNA. So, by methyling its own DNA to the same recognition sites, right. So, that is very important, the methylase or the methyl transferases activity is going on to the same recognition site against which the restriction enzymes are acting to target the foreign DNA. So, same DNA sequences, right. So, the recognition sites or the sequences are same. For the same sequences against which the RA is active for the foreign DNA, that is also susceptible by the restriction enzyme action, and so that the methylases is only modifying the same recognition sites, and this bacteria is going to create a very good distinguishable mark, and that will distinguish the DNA, its own DNA from the foreign DNA. So, you can see the same recognition sequences are present in the host DNA in bacteria, or its own DNA is having the same recognition sites. The phage DNA is also showing the same recognition sites, but they are protecting the same sequences by addition of methyl group. They are modifying the base and they have lost. So, this RE enzyme will not no more able to recognize this site with this modification, and how this can be protected from the REase enzyme. So, this is a kind of like epigenetic modification, it is like a evolution allow, and this is a kind of how it will allow the restriction enzyme to differentiate between the self and non-self DNA. Otherwise, there is a if it start in indiscriminate manner, you can see that restriction enzyme will also start cutting off the self DNA. So, this modification system will ensure to differentiate between these two DNA and ensuring that only foreign DNA is targeted for the cleavage, where the genetic material of the bacteria remains unharmed. So, it is now we have to see that once we started to study about the RM system, we found that that is not like, it is uneven distributed in the bacterial system, it is not universal in all in all species of the known bacteria. Approximately 1/4 of the known bacteria has been reported with this RM system, and we found like there are multiple RM systems, right. So, as we started to discover about the RM system, we found yes, there are different type of RM systems are there, and only 1/4 of the known are have possess this RM systems. So, the diversity in the RM system highlight the significant, like it is a kind of evolutionary pressure that how the bacteria are developing their defense mechanism to protect themselves from foreign DNA material, and we can say that among the bacteria RM system have been extensively studied, and they have been found reported with the wide range of the taxonomic genera, and some of the known species which I have shown in this slide of RM systems are very known bacterial species, like you can see Escherichia coli, Bacillus subtilis, Streptococcus pyogenes, Haemophilus influenza. These are the some of the examples which I have shown over here, and these are the types of the RM system. So, as per the, it is important, like as per their discovery in the chronological order, we have reporting like different type of RM systems are there based on their subunit protein structure, based on their cleavage sites, based on their recognition sites, based on their substrate specificity, enzyme complexity, energy requirement, their specific cleavage sites. You can see multiple type of RM systems are there. So, mostly you can see there are 4 types of RM systems are there, and this is they have been named as per their chronological discovery, and now we will see one by one the 4 types. So, this is the type 1 RM system. So, in this you can see that it is the heterooligomeric complex, it is having 3 important units in 3 different protein complexes. The one is the restriction, another is called to be the restriction is called to be the cleavage site, and there is a recognition site, and then there is a methylase site, right. So, you can see there are 3 important units are there in this enzyme. One is called as restriction, modification or methylation, and there is a recognition sites, and you know that this is the energy requiring process, they require ATP for this kind of activity for to cut the DNA, and they also need the S-adenosyl methionine as cofactors to start the catalysis, because this is the group S-adenosyl methionine act as a methyl donor. So, what it will do, it is having the 3 important component, restriction and modification we know. The other important is the specificity unit, that is also called to be the recognitions. So, the specificity unit or S unit will confer, HSDS will confer the specificity to the restriction and modification, that they are very specific for their modification site, for their cleavage site, right. And upon introduction to the foreign gene, this enzyme will first modify its own DNA, and then it will start recognizing and cleaving the foreign DNA. So, only this the complexity, it is a kind of complex enzyme, it is having 3 complex, right, and they work one by one, and thus they are 3 important subunits, and it can be called as a heterooligomeric complexes, and the requirements are like they need the ATP and one important cofactor. Coming to the next type of RN system. So, here you can see that this is very important to understand, like as I told you, they also differ in their recognition site and in their cleavage site. So, this is important in this type 1 restriction enzyme. Normally the restriction site is not in the, it is within the recognition sequences, they are actually from the considerable distances from their recognition site. Mostly they are 100 base pair or 10 of 1000 base pair upstream or downstream away from the recognition sequences. So, they can read in backward or in the forward direction. So, you can see here the recognition site is not the restriction site. Recognition site is different from the restriction site or the cleavage site. It is normally you can see considerably, you can see the distance, at variable distance the cleavage site is located. So, this is very important for the type 1 enzyme, the cleavage site and the recognition sites are not same, right. Coming to the this is showing the example one of the Bacillus subtilis type 1 restriction enzyme. As I told you, they are extremely diverse group of enzymes, and you can say that mostly the RM restriction modification enzymes are 4,000, more than that 4,000 the number of enzymes have been studied, and they have 3 different types of like specificities are there. So, we are taking some of the examples, like this is the Bacillus subtilis type 1 enzyme, this is the recognition sequence CTC, GAG is this, and the methylase the adenine base in the sequence. So, we have studied it, right, and methylation obviously will prevent the host DNA from the cleavage by BSU M1 type 1 restriction enzyme. Coming to the next, this is the type 3. So, I have skipped the type 2 because type 2 is one of the most widely studied one, right, and it is most prevalent, very common and very simple enzyme are type 2. So, before that I am taking the type 3 RN system. It is having a very we can say it is a hetero trimer or hetero tetrameric type of enzyme. It is also a complex enzyme. It contains both these functions, as we see the restriction and modification. The requirement is same, but you here see the difference in the recognition side. So, the complexity type 1 has having the additional S unit, which is not present in the type 3 RM system, and the recognition site is different from the restriction or cutting site. Normally, it is 25 to 27 base pair towards the downstream, 3 descent from the recognition sequence. So, they are in the downstream region, they are in the 3 region. So, they read only the backward. So, they will recognize their sequence, which has been go to the next slide. So, we will see here, you can see the illustration that this is the type 3 recognition enzyme. You can see here it is a recognition site, and once you see the cleavage or the cutting site is downstream to the recognition, and how much far? It is almost 25 to 27 base pair away from the recognition sequence. It is in the downstream region. So, it can translocate on the DNA molecule, and it will go downstream and it will start cutting from there. So, this is the example which is present in the Salmonella typhimurium. It is recognizing the specific DNA sequences, and we have so many such example in general which has been studied for the different type of enzymes. So, this is showing one of the example of type 3 restriction modification system of Salmonella. And now coming to the type 2. So, type 2 restriction modifications are one of the simplest and most prevalent, most widely studied and widely applicable enzyme also, because it is simple. Normally, we saw that they have the multi-unit complexes, the enzymes are type 1 and type 3 are multi-unit. Here you can see it is having like one single important, it is like a simple enzyme, and it is having 2 different polypeptides, right. One polypeptide is of restriction enzyme, and second protein subunits are like of modification. So, restriction endonucleases is one protein and modification is another protein. They are 2 separate proteins and they act independently, right. So, this is like they have number of advantages over 1 and 3 type, because they first cleavage and the methylations are catalyzed by separate enzyme, right. So, they do not have the single complex to act for the cleavage and methylases, they are having the different enzymes for the cleavage and different for the methylation. So, make the possibility to cleave the DNA even in absence of modification, right. So, the DNA can be cleaved even if it is unmodified, and they do not require ATP and SAM as cofactors. The restriction endonucleases of this type cleaves the DNA, as you know their recognition sequences are very specific, but what is important here, that this recognition sequence of the type 2 restriction and endonucleases are also 4 to 8 base pair long, palindromic, right, and the DNA methyl transferases of restriction type 2 also methylate the same recognition sequences. So, this is the highlighting feature of type 2 enzyme, that the same recognition sequences are methylated and where the restriction endonucleases are cleaved. But apart from that, we can say that methylation, as you know, the methylation will protect the DNA from its own self destruction, but this modification and the other important noticeable feature of type 2 is that the cleavage sites are near, or it is close to the recognition sequence, or it will be within the recognition sequences. They are not far from the cutting site is not situated far from the recognition sequence. The recognition sequence itself is the cleavage site, right, and it may be sometime often it can be close, but mostly you can see the cleavage site is same as it is recognition sequences. So, this is very important enzyme, right. It is particularly in the aspect of DNA repair and the DNA gene regulation, it is also functioning, not only protecting against the foreign DNA. So, this is the example. EcoR1 is one of the known type 2 enzyme. It will recognize a specific DNA sequence and it will cut the DNA, and this is the EcoR1 recognition sequences, which is a palindromic type. So, type 4 is just now, as I told you, because of the discovery, now we have identified one more type of restriction modification system. They are distinct from the remaining 3 groups. Here what is important and very peculiar, that it will cleave the DNA substrate when they contain the modified bases, like they will cut only the modified DNA, where you can see that either the methylation or hydroxymethylation or glucosyl hydroxymethylation has happened. So, why it is happening like this? As you know that as when the bacteria is evolved to defend itself from the viral DNA, at the same time, right, the viral is also finding the way how to evade this restriction modification system. So, what they are doing, like the host DNA is protecting itself from modification, virus is also doing the same thing. Once it attacks, it is doing some kind of modification, like either it is methylating it or hydroxymethylating, or they are making some modified bases, so that the restriction enzyme will block on those sequences. As the host DNA is protected, similar to that, the virus DNA is also evolved to protect its DNA from the restriction enzyme of the host. So, this has happened. So, virus is evading the restriction modification system. It will like restricting the restriction enzyme for its own attack. So, now what is important? Now, the bacteria at the same time, in turn, or to counteract the viral modification, bacteria also is expressing the modified enzymes, and this modified enzyme is one of the example is the type 4. So, remember, everything in nature is evolving. If the bacteria is protecting itself, virus is attacked, and viruses want to have the safe mechanism, so that it can be protected within the host cell. So, they are evading the restriction system, and to counteract, the bacteria is also having a new type of restriction modification system, and this restriction is only the cleavage. It will cleave the DNA which is only modified. So, what happened in the type 4? This unique feature is setting it apart from the other type of restriction modification, which will typically recognize and cleave the unmodified DNA sequences, but this type 4 enzyme will only act on, or it will cleave only the DNA substrates which are showing the modified bases. So, this modification, if the virus is successfully modifying the bases, then what will happen? This restriction type 4 system will attack the modified bases of the DNA of the virus, but your host DNA remain unmodified, right, unmethylated, it we cannot say right now, but this is called to be like unmodified DNA sequences. So, the host DNA is unmodified and the viral DNA is modified, and due to this modification, type 4 restriction enzyme will only act on the modified bases, and it will cleave the DNA substrate which specifically shows the modification in their nucleotides, like methylation, hydroxymethylation or glucosyl hydroxymethylation of the base at some specific sequences. So, this is a kind of a new reported type 4 RM system which is associated with the epigenetic regulation in the prokaryotic organism. So, this is a prokaryotic primitive immune defense. Now, coming to the how it can be useful. So, we know these are having a very important, these are naturally present in the bacteria as their defense mechanism, but they have been very well reported in the application. These enzymes are one of the very important tools in the DNA cloning, right, particularly the type 2, because it is a simple, they are very oftenly used to cut the DNA at some specific recognition sequences that will produce a sticky end. As I told you, once the restriction enzyme cut the DNA, they may produce blunt or cohesive ends, and with this ends you can ligate the foreign DNA and you can generate the first recombinant DNA, which is important for the cloning of the DNA sequence to plasmids or any other vectors. So, this restriction enzymes are naturally there as a bacterial defense system, but they have very good application in the DNA cloning. So, this is the one of the important foundation of the DNA cloning techniques, so that we are now able to cut the DNA to ligate the new DNA to the genome of the vector. So, we can introduce the foreign DNA, because we have to open the DNA to insert the new DNA, and this opening can be done with the help of the DNA cutters, and these cutters are basically the restriction enzyme. So, you can introduce the new genes to the host organism, and for various purposes of the genetic engineering, it may be the expression, protein production, or any other applications of the genetic engineering. So, this is how the plasmids, as you know, being a part of the genetic engineering, you can see that plasmids are also having multiple cloning sites, right. The vectors show the multiple cloning sites. Basically, these are the restriction sites, recognition sequences for variety of the restriction enzyme, and this is also called to be the cloning sites. Why? Because through this site we open the plasmid DNA with the help of the restriction enzyme, which creates the sticky end or the blunt end, to which we add the new DNA or the foreign DNA, and we seal the DNA with the help of the DNA ligase, and this is how firstly we create the recombinant vector DNA. So, this is about its application. So, I hope this will give you the substantial understanding and comprehensive understanding of the restriction modification system in general as well as its applied part. Thank you.